The next section of the anatomy review deals with the issues of cell biology and histology. And we're going to start out by looking at an individual cell with regard to its subcellular organelles and its cytoskeleton, and finish up this chapter with a comparison of various different forms of epithelia. Now, there are a couple of issues with regard to the anatomy review of histology. First off, histology is an all-encompassing subject. Aside from the fact that we're going to spend some time dealing with normal tissue identification, to review a functional histology implies that we have to talk about biochemistry, physiology, pathology, pharmacology, and so on and so forth, which obviously we're not going to get into. So we really want to concentrate on normal tissue identification. Why does a tissue look like it does? And when we deal with a tissue, recognize that there are four fundamental tissues that are going to make up virtually every organ in the body. Every organ basically has epithelial tissue, connective tissue, muscular tissue, muscle, and nervous tissue. So our job as we go through this is to identify those tissue components, deal with what cell types might be present, what their functional significance is, and particularly when we get to epithelia, exam questions frequently deal with where are the stem cells in a tissue or an organ or an epithelium, and what are they actually stem cells for? All right, the other issue we're going to deal with when we get into cell biology, which is first, and that is, what are the different functions and the appearance of organelles? And histologically, we can kind of subdivide organelles into either membrane-bound organelles, which will be the largest of the subcellular structures, versus non-membrane-bound organelles, and non-membrane-bound organelles make up the cytoskeleton. So we're going to distinguish those not only anatomically functionally, but what we want to concentrate on is what cellular components, membrane or non-membrane bound, look like in the electron microscope, which is frequently how they're viewed. All right, so the first organelle we're going to deal with is the largest of the membrane-bound organelles, and this is the nucleus. Certainly, as you know from biochemistry, it's the site of DNA replication and transcription of DNA into RNA molecular precursors. Contains all the enzymes required for repair and replication of newly synthesized DNA and its transcription. Nucleus is one of two organelles that's actually bound by a double membrane in the form of the nuclear envelope. And inside the nuclear envelope will be the nuclear lamina, a nucleolus, basically what we call a nuclear inclusion, and obviously two different forms of chromatin. The nucleus itself in the electron microscope, like everything in the electron microscope, is either black or white. So when structures are described in the electron microscope, it's kind of as if you were interpreting a radiograph. Things are considered in the electron microscope to be either electron dense, which means they're black, or electron lucent, which means they're light or unstained. And this story kind of begins with a picture of a nucleus. That happens to be from a liver hepatocyte, although it could be from virtually any protein-producing cell. And you'll note first off that in the EM, the dominant feature of a nucleus is its chromatin content. And certainly in a typical active protein-producing cell, euchromatin is going to be much more prevalent than heterochromatin, because euchromatin is the active form of chromatin, and it constitutes in a typical protein-producing cell about 90% of the available chromatin, whereas heterochromatin, the more electron-dense form of chromatin, black, shown in this micrograph, is the inactive form. And this is also a very good electron micrograph of a nucleolus, as we well know, a site of ribosomal RNA synthesis and ribosomal assembly. The nuclear envelope, as we said earlier, is a structure that is in the form of a double membrane. And it contains pores that we'll see are continuous with the endoplasmic reticulum. And there's only one other subcellular organelle that contains a double membrane surrounding it, and that will be a mitochondria. Chromatin, as we indicated, it's either in the form of heterochromatin, which is transcriptionally inactive, constituting about 10% of the chromatin in a typical eukaryotic cell, or it's euchromatin, which is basically a more extended and active form, which accounts in a typical protein-producing cell for about 90% of the total chromatin. Next, the nucleolus. We alluded to this earlier in the electron micrograph. It's responsible for ribosomal RNA synthesis and ribosomal assembly. There are a couple of morphologically distinct zones which can be identified in an electron micrograph. The active zones are the granular zone and the fibrillar zone, one of them found at the periphery, one more centrally located. But the fibrillar center, the actual central portion of the nucleolus, is the part which is inactive in that it contains DNA that's not being actively transcribed. Now that we've talked about the nucleus, and the last comment we'll make about it is the following: virtually every eukaryotic cell has one, except for red blood cells, but recognize that there are certain cells that have more than one nucleus. Hepatocytes would be one of them. They're frequently binucleate. Osteoclasts, which we'll see when we get into bone marrow, they're typically multinucleate, as are megakaryocytes. All right, the other membrane-bound organelles that we're going to spend time on, functionally as well as anatomically, are the elements of the endoplasmic reticulum. It comes in two flavors, rough and smooth. One of those, the rough ER, is closely associated functionally with the Golgi apparatus. And the Golgi apparatus has some functional relationship to lysosomes, which we'll see. But we'll distinguish lysosomes from peroxisomes or microbodies, and finally all of those from the other double membrane-bound organelle, which are the mitochondria. All right, so first, before we do that, let's look at an overall schematic view of a typical eukaryotic cell, and it shows quite nicely the configuration of the nucleus. You can see forms of chromatin, schematically illustrated as being coiled up, heterochromatin, since you're visualizing it. You can see that the nuclear envelope is a double membrane configuration with pores, and you note that the nuclear pores are directly continuous with elements of the smooth and rough endoplasmic reticulum. Not labeled, although certainly prominent, is a nuclear inclusion, which is certainly the nucleolus, the little nucleus that we identified earlier. Now, we're going to proceed by talking about the elements of smooth versus rough endoplasmic reticulum. You can see in this schematic configuration that they consist of parallel arrays of membrane-associated cisternae. The difference in them anatomically is the rough ER is characterized by the presence of electron-dense dots attached to its membrane components that make it rough, and the dots basically, as we'll see, are bound polysomes. The smooth endoplasmic reticulum looks very much like rough endoplasmic reticulum, but without the electron-dense dots. Next, note that smooth endoplasmic reticulum, particularly in this picture, looks very similar to the Golgi apparatus. Very different functionally, but we're going to have to distinguish them very carefully in different electron micrographs, and we'll do so. Then, certainly the smaller of the membrane-bound organelles, also illustrated in this picture, are mitochondria, lysosomes, and peroxisomes. And then finally, this also gives us an indication of what some of the cytoskeletal elements are. We have three flavors of those. Cytoskeletal elements come in the form of microtubules and two forms of filaments. One class are known as intermediate filaments. The other class are known as microfilaments. And then, although not labeled on here, a couple of the other features of cell biology we need to discuss are how cells are related to additional cells by junctional specializations, and finally, how cells have certain luminal surface specializations of a wide variety of types that underlies a different functional specificity. Next conversation deals with ribosomes. Ribosomes are composed of ribosomal RNA and protein, and they have a combination of large and small subunits. They are assembled in the nucleus and transported to the cytoplasm through the nuclear pores. Frequently, the ribosomes will form polysomes, which consists of a single messenger RNA that's being translated by several ribosomes at the same time. The important point about these polysomes and ribosomes, they exist in two forms. The bound polysomes are those that are attached to and functionally associated with the endoplasmic reticulum. The free polysomes are situated in isolation in the cytoplasm and not associated at all with the endoplasmic reticulum. But here's the point. recognize that free and membrane-associated or bound polysomes each produce three different kinds of proteins. Arguably, the more important array of proteins are produced by the membrane-associated or bound polysomes that are associated with the endoplasmic reticulum. And important because those three types of proteins are secretory proteins, membrane-associated proteins, and lysosomal hydrolase enzyme proteins. The proteins produced by free polysomes all stay within the cell, unlike some of those produced by the membrane-associated ones. And there are three different proteins produced by these unbound polysomes. They are those that are to be incorporated into the nucleus, incorporated into peroxisomes, or are associated with mitochondria. Next, what does rough endoplasmic reticulum look like in the electron microscope? As shown well in this particular micrograph, it is a series of parallel arrays of cisternae that have the same diameter end to end. In other words, the form of them is the same end to end. They are parallel, and again, they're easy to recognize because they are characterized by the presence of electron-dense dots, which represent membrane-associated polysomes. Smooth ER, on the other hand, looks like rough ER, but lacks the bound polysomes or ribosomal association. And one of the functions of basically smooth endoplasmic reticulum, its major thing, is to produce steroid-based hormones, but it's also associated with a wide variety of other biosynthetic reactions. So first off, let's compare the two. Note that in this electron micrograph, note how regular and uniform the structure of rough endoplasmic reticulum is. Not only are the cisternae basically the same diameter end to end in an individual cistern, but note that the cisternae are almost equally spaced from one another in this electron micrograph. In contrast, smooth endoplasmic reticulum has a much more irregular array of its cisternae, nowhere near matching the regular pattern that's seen in the adjacent rough endoplasmic reticulum. And using the pointer, we can see other membrane-associated organelles. This happens to be from corpus luteum. So the mitochondria have a kind of a Christie-like tubular matrix in them. So these are all membrane-bound mitochondria down here. The electron-dense structures could be microbodies, could be primary or secondary lysosomes. It's kind of hard to tell based on their configuration without specialized staining. Now, what else is smooth ER doing? We identified its involvement in steroid hormone synthesis. It's also involved in hydroxylation reactions, detoxification reactions, mainly by using a whole array of enzymes that functionally are associated with cytochrome P450. And in addition, smooth endoplasmic reticulum is also involved in conjugation reactions. Lastly, it's involved in glycogen metabolism, involving degradation in gluconeogenesis, lipid metabolism, and as we'll see in striated muscle cells, skeletal and cardiac muscle, it serves as a sequestration site for calcium ions that are closely associated with the cell membrane of skeletal and cardiac muscle cells. Next, in this electron micrograph, we have a view of the Golgi apparatus. And note that Golgi apparatus looks very similar to smooth endoplasmic reticulum in that it consists of parallel arrays of cisternae. The difference in the electron micrograph is that the Golgi apparatus typically has a slight curvature to it. So it's going to have a convex face and a concave face. And second, at the ends of the cisternae, the parallel cisternae, frequently we see, and we certainly do here, dilatations or blebbing on the ends. So unlike the smooth endoplasmic reticulum, if it were an irregular pattern, the Golgi apparatus cisterns do not have a uniform diameter from end to end. Now, what does the Golgi apparatus do? Well, it is closely associated with the rough endoplasmic reticulum, because each of the three kinds of proteins that are produced by the rough endoplasmic reticulum must next be sent to the Golgi apparatus. And you can see the proximity of the cis face of the Golgi apparatus. That's the entry point of all three of the proteins newly synthesized by the membrane-associated polysomes of the rough endoplasmic reticulum. So the medial Golgi is a site where those proteins begin the sorting and glycosylation process. And by the time the proteins reach the trans face, the packaging of them has occurred after sorting. And if they're going to be packaged for secretion, typically the newly synthesized protein will be incorporated into a membrane-bound secretory vesicle that will then move and fuse with the extracellular membrane of the cell, and the secretory granules be extruded by exocytosis. Now, two clinical scenarios basically involve the Golgi apparatus. One is hyperproinsulinemia. This is characterized by elevated levels of proinsulin in the serum because of an enzyme defect, a peptidase defect that fails to cleave proinsulin to insulin and a related C-related peptide in the Golgi apparatus. And the clinical manifestations of this are similar to patients seen that have non-insulin-dependent diabetes mellitus. The other Golgi-related disease is known as inclusion cell disease or eye cell disease, named for dense inclusions that typically are present in fibroblasts. This eye cell disease is again caused by an enzyme defect. Specifically, it's a phosphotransferase enzyme defect that fails to adequately mark hydrolase enzymes for incorporations into lysosomes. Because as part of the sorting and marking process, those hydrolase enzymes have to be phosphorylated. And the phosphate basically is added in a two-step process that involves that phosphotransferase enzyme. That enzyme typically is deficient, so what in essence happens is there's tremendous inclusion bodies that occur in fibroblasts, and basically the lysosomes lack their appropriate hydrolase enzymes. Clinical features of this involve multiple skeletal abnormalities and a lifespan of typically less than 10 years. Next, speaking of lysosomes, lysosomes are our next membrane-bound organelle, and as such they contain the very low pH hydrolase-based enzymes that are required for intracellular digestion of large macromolecules. Lysosomes generally come in two forms, although the distinction between them is not as precise as it used to be. Classically, a primary lysosome has not acquired materials to be digested and has just been formed by budding from the trans face of the Golgi apparatus containing its hydrolase enzymes. A secondary lysosome is basically involved in some aspect of the degradation process and typically has some sort of interiorly located, partially degraded substrate. Lysosomes comprise or contain about 60 hydrolytic enzymes. All virtually are acid hydrolases, but again, these hydrolases are produced in the rough endoplasmic reticulum. They have to be transferred to the Golgi where they are modified by phosphorylation, sorted, and packaged ultimately into lysosomes. And as you'll hear in pathology, there are a number of lysosomal-based storage diseases based on various hydrolase enzyme deficiencies that result in deficient breakdown of sphingolipids in the clinical conditions known as Gaucher's, Niemann-Pick, and Tay-Sachs disease. Finally, an electron micrograph view of primary and secondary lysosomes. Probably the homogeneous electron-dense structures toward the middle of the picture are most likely primary lysosomes, and the structures at the tip of the arrows are more likely secondary lysosomes that contain partially digested substrates. Now, how do lysosomes differ from peroxisomes? Peroxisomes are really a specialized kind of a lysosome in a way. They're frequently referred to as being microbodies, and like a lysosome, they contain a number of enzymes that are involved in various stages of degradation. But in this case, the major product, toxic product that's being degraded by peroxisomes is hydrogen peroxide. And the major enzyme, therefore, that's a component of peroxisomes is catalase. Specifically, its job is to degrade the hydrogen peroxide to water and oxygen. But peroxisomes are also involved in the oxidation of long-chain fatty acids, phospholipid exchange, bile acid, as well as cholesterol synthesis. And peroxisomes are unique in that their enzymes, including catalase, as we said, are proteins that are basically produced by unbound or free polysomes within the cytoplasm. These enzymes that end up in a peroxisome do not have to pass through the Golgi apparatus. They're simply incorporated directly into the peroxisomes. But peroxisomes may lack one or more of their catalase-based and other enzymes, giving rise to a number of peroxisome biosynthesis disorders, or PBDs, the most severe of which gives rise to what are called empty peroxisomes on exam questions. And this is Zellweger syndrome. And the mildest form of a peroxisome biosynthesis disorder is Refsum disease. And again, you'll distinguish some of the signs and symptoms of these in pathology. Finally, we see a view of a peroxisome. It's kind of difficult without specialized staining techniques to visualize. It's kind of shown here on the right side of this electron micrograph. But it makes a good comparison between a membrane-bound kind of amorphous and relatively homogeneous structure, such as a peroxisome or a primary lysosome, compared to that of our last membrane-bound organelle, and these are mitochondria. Mitochondria, like the nucleus, are the only other subcellular organelles that contain two membranes. And certainly mitochondria are very distinct in that their inner membrane is thrown into folds known as cristae. And certainly the major job of mitochondria is to synthesize adenosine triphosphate, or ATP. They are capable of making their own proteins, and they have their own DNA as well. And it's important to recognize where the ATP synthesis and the oxidative phosphorylation are taking place. First, on this picture we've shown before, we can visualize quite nicely the Golgi apparatus, but although we didn't allude to it before, you can see multiple examples of the hot dog-like structures known as mitochondria, and you can see how evident and obvious the internal cristae matrix of the interior is. Note again that the real business end of a mitochondria is its inner membrane. It's thrown into numerous foldings or infoldings called cristae that dramatically increase the surface area of a mitochondria, and they are the actual sites that contain the enzymes associated with electron transport and oxidative phosphorylation. And one clue as to the metabolic activity of an epithelium or a tissue is based on the presence of not only the number of mitochondria, but the number of cristae per mitochondrion, which is largely proportional to the metabolic activity of the cells in which they reside. Now let's move our conversation from the membrane-bound organelles to the non-membrane-bound organelles, which is really a fancy term for the elements that comprise the cytoskeleton. Note that the cytoskeleton consists of three different elements. The smallest of them are microfilaments, consisting exclusively of actin. Kind of intermediate in size, hence the name intermediate filaments, are tissue-specific filaments that consist of a number of different proteins that provide a structural integrity to cells in a wide variety of organs. Lastly, the largest of these cytoskeletal elements will be the microtubules, and we'll see a variety of locations in which we find them. First, let's start with microfilaments. Microfilaments consist exclusively of actin proteins that basically are globular monomers of G-actin that have polymerized to form helical filaments of F-actin. The actin polymerization is energy dependent. They typically are about 7 nanometers in diameter, and they have a distinct polarity. The barbed end is the site of polymerization, and the pointed end depolymerization. And treadmilling is in balance and activity at both ends. And one of the reasons this is important is that microfilaments convey a motility to cells, particularly cells that are actively moving in and out of the bloodstream, such as white blood cells. But in addition, microfilaments will form a contractile ring that will be used in cytokinesis of cells undergoing mitosis or meiosis. And lastly, Microfilaments, actin proteins, will form the core of a cell surface specialization known as a microvillus. Next, what about intermediate filaments? They are consisting of tissue-specific proteins, and overall they convey a structural integrity to a particular cell. And there are a couple of different types of intermediate filaments. Type 1 consists of keratins, and keratins basically are found in all epithelial cells, whether they are ectoderm, mesoderm, or endoderm derived in terms of embryology. Type 2 have a fair diversity of them. Desmin is a type 2 intermediate filament that's found in virtually all muscle cells, particularly skeletal muscle cells, cardiac muscle cells, and at the very least GI tract smooth muscle cells. Vimentin, another type 2 intermediate filament, is found in fibroblasts, endothelial cells, and blood vessel or vascular smooth muscle. It forms what is known as glial fibrillary acidic protein, which is found in glial cells such as astrocytes and most Schwann cells, and peripherin, which is an intermediate filament that maintains the structural integrity of axons in the peripheral nervous system. Type 3 are intermediate filaments that are found actually forming structural supports in the cell bodies and dendrites of neurons. And type 4 intermediate filaments form lamins that form a meshwork inside the nuclear envelope of all cells. Finally, microtubules. Microtubules are in essence hollow tubes. They are the largest of the cytoskeletal elements, and like microfilaments, they are undergoing continuous assembly and disassembly because one of their functions is intercellular transport of vesicles, molecules, and they're particularly important in the nervous system because they are the subcellular organelle that's used in axonal transport in both the anterograde and the retrograde direction. Transport, regardless of where it is an energy-requiring process, and dynein drives what's known as retrograde transport. Kinesin drives the component of anterograde transport in neurons as well as any other cell type that utilizes microtubules. So, in addition to providing vesicular and molecular transport inside cells, microtubules are the cytoskeletal element that are found in true cilia and flagella, and as such, because these are motile structures, they utilize dynein to maintain the beating nature of true cilia, and they all obviously are found in the tails of sperm cells, allowing them motility as well. And then finally, not the least of which, is microtubules form the mitotic spindle, which is going to help during chromosome separation during mitosis and meiosis. All right, one of the things that affects intermediate filaments, which is particularly relevant in the nervous system, is Alzheimer's dementia. But we also see it in cirrhotic liver diseases as well, that basically are caused by initially changes in the intermediate filaments. Colchicine prevents microtubule polymerization and is used to prevent neutrophil migration in gout. And there also are elements, vinblastine and vincristine, that are used in cancer therapy because they inhibit the formation of the mitotic spindle of dividing cancer cells. Next little topic we need to address, because their importance in pathology as well as functional histology, are the cell adhesion molecules. And there are two classes of them. Cadherins and selectins are adhesion molecules that are calcium dependent, whereas integrins are adhesion molecules that are calcium independent. And basically, in one form or another, these classes of cell adhesion molecules are associated with intercellular junctions. Speaking of those junctions, let's look at the diagram on the next page, because this is our next topic. We need to distinguish between the different forms of junctional specializations that either unite adjacent cells, particularly epithelial cells, to each other or with each other, or unite the basal lamina of the underlying epithelium to the epithelial cells themselves. So as we'll see, we're going to be dealing with a number of junctional specializations. So let's review their anatomy and location now, and then we'll talk about their other additional features. First off, tight junctions, or zonula occludens junctions, are found only at the luminal surface of an epithelium. and they, in essence, as we'll see, serve as a barrier. They have punctate points of contact, sealing off the extracellular space between the opposed membranes, which are shown schematically in this picture. Directly below all tight junctions or zonnular occlusions are zonnular adherens. Zonnular adherens junctions help maintain the structural integrity of the zonnular occludens, and these junctions have a slight electron density in the adjacent cytoplasm of the opposed cells. The density is formed because of the interaction between the membranes at the zonnular adherens and microfilaments that consist of actin. So in summary, both the tight junction, even though it's not evident on this picture, and the zonnular adherens are closely associated with the microfilaments in the apical part of the cytoplasm of adjacent epithelial cells. Underlying the zonnular adherens, you can see a couple examples of desmosomes. Desmosomes are junctional specializations that help keep epithelial cells together. So they're going to be particularly prominent in an epithelium that's under considerable stress, such as the stratified squamous epithelial cells that make up the epidermis. But unlike the first two junctions, desmosomes are closely linked with intermediate filaments, particularly keratin intermediate filaments in an epithelium in the adjacent cytoplasm of the joined epithelial cells. So desmosomes obviously are playing an additional role in addition to the intermediate filaments in maintaining the structural integrity of the cells they are associated with. Now, a specialized form of a desmosome is a hemidesmosome. And the job of a hemidesmosome is to keep the corresponding epithelial cell closely anchored to an underlying basement membrane or basal lamina. Lastly, gap junctions. Gap junctions promote cell-to-cell communication. And basically they consist of a series of pores formed by connexins that are lined up between the adjacent cell membranes. But unlike the other intercellular junctions, gap junctions are not associated with any cytoskeletal element. All right, so here are the details. As we said, a zonnular occlusion functions as a barrier to basically the extracellular space. It helps determine cell polarity. The major components of that barrier are occludins and claudin proteins. The zonnular adherens lies directly below the zonnular occludins. It helps maintain the integrity and the proximity of the zonnular occludins. And this is where we find cadherins linking the adjacent membranes. And as we said earlier on the previous slide, both of these junctional specializations are associated with actin microfilaments in the adjacent cytoplasm. And cadherins have tremendous pathological significance because a first step in the invasion of malignant cells through an epithelium basically results from a loss of expression of those cadherins, linking the zonnular occludins of adjacent cells and thereby weakening the epithelium. And next, gap junctions in no particular order are the communicating junctions. They function in cell-to-cell communication. They consist of small arrays of connexin proteins that line up to form a connexon, which is a tubular channel through which small ions such as calcium and small low molecular weight molecules such as second messengers, including cyclic AMP, may pass. And as we said earlier, gap junctions are the only junctional specialization that are not associated with any cytoskeletal element in the adjacent cytoplasm. And then desmosomes. Desmosomes, synonym for them, form spot junctions, also known as immaculate adherens. They help maintain the structural integrity of an epithelium by providing a mechanical link between the cells. Cadherins here as well link the adjacent membranes. And as we know, the cytoskeletal element that's closely associated with a desmosome are intermediate, usually keratin intermediate filaments in the adjacent cytoplasm. And hemidesmosomes, as we said, link epithelial cells to the underlying basement membrane, but instead of utilizing cadherins as their adjoining molecule, they utilize integrins that link hemidesmosomes to the underlying components of that basement membrane. Finally, a couple of pictures. This is a freeze fracture, one way of studying intercellular junction, and freeze fracture of a zonnular occludens or a tight junction has a very distinct array of what we call sealing strands that are kind of irregularly situated, but they represent the punctate points of contact, sealing off the extracellular space by providing a closure mechanism linked by claudins and occludins at the level of those sealing strands. And finally, an electron micrograph that illustrates the locations of a couple of these junctions. And again, you can classify these junctions based on their location as well as their form and function. Note that a zonula occludens is virtually always at or near the luminal surface of an epithelium. And even though structurally it's associated with actin microfilaments in the adjacent cytoplasm, a zonula occludens in a typical electron micrograph such as this looks like an electron-dense line. And situated below a typical zonula occludens is a zonula adherens, both of which are associated with actin microfilaments. The actin microfilaments tend to be a little more prominent at the zonula occludens, giving them a slight intracellular density, electron density, on the side of the opposed membranes. Now, in this example, directly below the zonula occludens, but is a desmosome. Desmosomes can be found basically between any part of an epithelial cell, pair of epithelial cells. We happen to show this one directly below the zonula occludens. But note the significant intracellular density on either side of the adjacent membranes. Desmosomes are very easy to recognize because of the presence of this density, and the density is so prominent because, unlike the zonula occludens and the zonula adherens, the desmosome is linked to intermediate filaments in the adjacent cytoplasm. All right, last but not least, what are the luminal surface specializations that might characterize various epithelial cells? Well, fundamentally, there are three kinds of cell surface specializations, but one specialization is really a subset of another. So really, when we deal with luminal surface specializations of epithelial cells, we are talking about either microvilli or true cilia. So if I go back to the previous page for a moment, we can see that this particular micrograph shows a series of microvilli. These are luminal surface specialization that contain a core of actin microfilaments. And their job fundamentally is to increase the surface area of an absorptive epithelium. So because in this example of the presence of the glycocalyx, it's likely that these two epithelial cells are some of the lining enterocytes that are highly characteristic of much of the small and large intestine. So, as we say, microvilli contain a core of actin microfilaments. Their job is to increase the absorptive area of an epithelial cell. As we just showed in the previous example, they are typical of the columnar epithelial cells of the intestine. They are characteristic of the cells in the proximal tubule of the kidney, and they're also basically found in the columnar epithelium of the respiratory system. So a specialization of microvilli are known as stereocilia. These are tall, usually branched microvilli that are found in a very restricted location, typically only found in the epithelium of parts of the male reproductive system, specifically the epididymis. So this is where they are branched. If there are unbranched stereocilia, they are characteristic typically of all of the inner ear hair cells in the vestibular and cochlear labyrinth. So stereocilia basically are simply tall microvilli. They certainly have an absorptive function, we think, in the epididymis, but they form the hairs of hair cells that basically are sensitive to fluid displacement. And when the stereocilia bend, they obviously exact changes in the underlying membrane potential of those hair cells. Lastly, how are true cilia different than microvilli? And the answer is true cilia are larger because unlike microvilli that contain a core of actin microfilaments, true cilia contain pairs of microtubules, typically in the arrangement where there are 9 peripheral pairs of microtubules that surround a central pair of microtubules. And unlike microvilli, microtubules and the cilia that contain them are motile structures because the cilia utilize an ATPase motor molecule known as dynein to allow the microtubules to glide against one another, allowing the cilia to bend and beat on the luminal surface, particularly of respiratory epithelial cells. And as we've already pointed out, they form the core of the flagellum, the motile tail of a sperm cell. And electron micrograph on the left shows a nice comparison between the size and diameter of microvilli compared with the size and structure of a true cilium. Note that this is an epithelium probably from the respiratory system that has a combination of true cilia and microvilli because it is in part an absorptive epithelium. But note that the structure of a cilium, the size, is about 4 or 5 times greater than the size of an adjacent microvillus. But certainly when you cut them in cross-section, as in the insert at the top right of the micrograph, you can see the quite characteristic array of 9 peripheral pairs of microtubules surrounding a single central pair. And all cilia are anchored into what is called a basal body, labeled B here, in the apical cytoplasm. And certainly just for reference, since this is an epithelium, we see between these adjacent columnar epithelial cells an occluding junction that represents an electron-dense line, and just below that, basically a junction that's called an intermediate junction here. That's an old term. That's basically a zonula adherens with a more prominent density conveyed by the adjacent actin microfilaments. And then just the details of the substructure of a true cilium. Again, you see the array of microtubular peripheral pairs, 9 of them surrounding a central pair. And the key element is what we call the dynein side arms. They contain the ATPase motor molecule dynein that conveys the motility to allow the adjacent microtubular pairs to glide against one another, conveying a motility to a true cilium. So the clinical correlate associated with true cilia is Cartaginer syndrome. Basically, it renders the cilia and their microtubular pairs immotile, and if it occurs in males, it's going to render their sperm incapable of swimming and result in infertility. And in either sex, since the true cilia are a prominent feature of the columnar epithelium of the respiratory tract, these individuals are prone to chronic respiratory infections because they have a decreased ability to move mucus out of the respiratory system and into the pharynx for swallowing.